Well, greetings, everyone. My name is Liz Pierce, and I would like to welcome everyone to this week's session of our Integrating Bioinformatics Educational Series. This week, I am pleased to introduce Dr. W. Kelly Thomas. Dr. Thomas directs the Hubbard Center for Genome Studies at the University of New Hampshire. This center is a leader in comparative and environmental genomics with a special emphasis on novel model species. Dr. Thomas is interested in using molecules to understand relationships among organisms and between organisms and their environment. Dr. Thomas is also very well known for his work in bioinformatics education. Most recently, he has become focused on the need for broad-based effective training in bioinformatics and rigor and reproducibility. Based on the successful development of curriculum support models for New Hampshire INBRE, he and his colleagues have established a training workshop using the INBRE mechanism. This week-long workshop called Train the Trainer facilitates the integration of bioinformatics into the undergraduate curriculum. Dr. Thomas, thank you very much for joining our series today. We're looking forward to your talk.
No, thanks very much, Liz, and thanks to everyone for coming and for inviting me. As I was mentioning to Liz, I somehow am running our COVID testing operation, which is a 4,000 test a day process and involves 12 hours a day, 6 days a week. I never imagined the university would remain open this long. So when I originally scheduled this, I was assuming that I would have lots of rest, and that has not happened. So if it seems like I'm dragging a little bit, I am dragging a little bit. So I really am happy to talk to you about this. As Liz mentioned, this training in bioinformatics is something that I have taken on late in my career. And it's a natural outgrowth of genomics and training students, and so I'll walk through our process and the principles that we have for this, and please chat your questions as we go. I'll try to pause along the way as well. Oopsie, no, all right, I'm learning. So the outline for this, we'll talk about some background information and justification for the approach that we're taking, the specific approach to program support that we have, our experience in implementing this both in the context of our role as the bioinformatics core for our INBRE program, and then later on in our IPERT program, and then some things about publishing. So I think we're all probably aware, probably why you're here, is that we're facing this time now, and have been for a while, of big data. And nothing exemplifies this more clearly is the rapid growth of genomic data that's happened. And it's happened to us so fast that it's outpaced our ability to naturally develop the skills to handle this. And so there's this bioinformatics gap. If you look on the diagram, which is an old diagram, but it's still true, we went through this period of dramatically increasing data production. And if you really pay attention to the amount of time, what happened during that period of time is that it became almost impossible to work in this field and the fields that are driven by genomics, like proteomics and metabolomics and all sorts of other things. That's maybe an opinion, not necessarily shared by everyone. That period of time is less than a PhD. Well, at least it was for me. Maybe other people were faster, but that's so short. And to expect this natural, what has happened over the history in science of a pace that allows a natural development of curricula to follow scientific progress, that was just not possible at this time. And so what remains is a training gap, and not just a training gap of our students, but a training gap of our faculty and practitioners, including me. So what's the premise here? So first of all, that these bioinformatics skills or informatics skills, or however you want to define them, are really core and foundational to many areas of biology, and in fact to other areas in science. One of the most important parts, and somewhat unique to our approach, is recognizing that bioinformatics is practiced in an environment that is extremely terse, and it's that command line or bash environment that is dominated by the use of open source software that is run on servers. There are other ways of doing bioinformatics, and admittedly, but this is really where bioinformatics is done in the long term. And I'll justify that in a little bit. Okay. But it is something that is foundational and sort of unwavering in our program. So it's also that working in that environment is also the first hurdle that people face, and it's the one that turns back 90% of the people, because it's just annoying. And it's just enough to make people say, I don't like this, I'm not going to do it anymore. Or get somebody else to do it for them, or find some other workaround. Okay. However, if you can enter this environment successfully, then it opens up this vast and rapidly growing and rapidly developing set of tools that bioinformatics practitioners use to analyze data. And I strongly believe that this should be experienced early and broadly in an undergraduate curriculum context. We can talk about that more. I was first introduced, and some of you may be familiar with the phage hunters. I was first introduced to this several years ago now by the guy I highlighted there that was part of one of these papers reporting out those results from the development and implementation of that. D. Denver, who's a faculty member at Oregon State, I think he was my second graduate student. And at the time, this began early on, even preceded next generation sequencing when that program began. This was a program that was developed for undergraduate students, and although we do things differently, really the benefit and the potential benefit is really well documented in this paper. So the other thing that is driving the specifics of our program, which is heavily genomic based, is that access to DNA sequencing information and novel DNA sequencing results, which are part of our program, is made possible by the rapid reduction in cost. And what is possible, when phage hunters came on, phage were possible to sequence, and now for a fraction of the cost, when they started phage hunters to sequence a phage, you can sequence bacteria or fungi and more now as time moves forward. So when we first started this, it was in the context of trying to develop undergraduate curriculum support within our INBRE program. And we wanted to do something, because at the time we were spending a lot of time training individual cohorts of undergraduates engaged in undergraduate research. But what we really wanted to do was to be able to help our partners and ourselves introduce this into the curriculum so it was sustainable. And that meant engaging large numbers of of students, relatively large. I mean, many of us, are at institutions where the classes are still small, but introducing them to that early and for as many students as possible. So the idea was trying to develop this so that it might work in things like genetics or microbiology or even first-year biology. One of the other things that I'll try to explain in a little bit is that if we're going to do that, then this bioinformatics needs to sort of naturally integrate with basic principles of biology. And I think that, for example, some of the approaches that we have can actually do that. So it's not just learning, how to type commands on a computer, but you're actually doing analyses that can support basic understanding in biology. And then we probably won't get too much to rigor and reproducibility and data analysis, but that's a core to the activities that we have. And that it's active and discovery based. I will say in the age of COVID and switching to online classes, teaching bioinformatics is really fun because it's something that you can really do and have it be very active. I know that my people and I this summer had 3 full weeks of bioinformatics training with about 25 students each week in support of our INBRE program and my IPERT, which unfortunately we couldn't do in person. So why are we doing it this way? Well, we really early undergraduate research experiences, which we think this, even if it's done in the context of a class, because it's novel and it's really research. It's got this discovery element. And one of the other things that we learned really early on is that students actually know that bioinformatics skills are core to their workforce training and their CVs, basically. And so it's been interesting because many of the other things that I teach, they're mostly wondering, why in the world are you teaching me this? What is this going to do for me? But very many of them are really hungry for these skills. The other thing that was important in the context of our INBRE program was that the way that we set it up was trying to do it inside existing curricula, because I know, especially at our primarily undergraduate institutions, suggesting that they teach additional courses is not met with joy. However, modifying existing courses is enough work. But we can talk later about, since we've had some years of experience, how that position has changed a little bit. So I just want to touch base on one of these key issues, and that is the Bourne again shell, or this bash environment, right? And I'll make some fairly strong statements. And one is that this is where bioinformatics is done. So if you have a professional bioinformatics person, this is where they do it. It opens the door to that vast library of free open source software that is the backbone of virtually every bioinformatics analysis. It is how people deal. This environment is not just for bioinformatics, it's how and where people deal with all kinds of forms of big data. So these skills are transferable to all kinds of big data areas. The other thing about this is that while the programs that you run to do specific kinds of analyses, whether it's assembling a genome, doing SNP analyses, RNA-seq analyses, all these sorts of things, change from day to day. Any of you that are doing this, you submit a paper and your 3 versions later on the programs that you've used by the time it gets published and you're asked to redo everything. Unlike that, this environment is extremely stable and durable. So this environment and working in this hasn't changed in decades, more than incrementally. And so these skills will not change and will be stable for a long time. So, so that's the environment that we train in. We think that the training that I'm going to talk about works well inside of a biology curriculum, and that we think that we're giving people some core and durable skills. If there are any questions for that first part, great. Otherwise, I'll move on to the next is what our program actually, the one that we have and share supports. So in order to implement this, we as part of our New Hampshire INBRE program, we had to set up a curricular support structure. And so this involves primarily a set of instructional videos. We have 33 of them, and the vast majority of these are active participation. So these are YouTube videos, but they're not meant for you to just sit and watch, because we actually provide all of the students with access to the server that is pictured in these videos, and sample data that they can work through and to develop those skills. And so by doing this, this allows some opportunities for flipped classroom designs where you can assign people to do particular sets of tasks. These videos, I will say, are also used as prerequisites for a number of other more domain-specific or advanced bioinformatics classes that also work in this environment. It gives them a strong basis for that. So we provide on our servers here that are New Hampshire INBRE servers, the computer infrastructure support. And as I'll talk about a little later, we provide instructor support, worksheets, test materials, sequencing, and online help. So what is the outline of our program? So the fundamental thing that we started off with was much modeled after the phage hunters, was to collect pure cultures of bacteria or phage, or more recently, environmental samples for community analysis. So the way that we have run this is that basically we give each of the participants their own bacteria or their own sample from an environmental sample, like a microbiome sample, with the environment in the broadest sense. So this then requires, or one can skip some of the molecular biology, so extracting DNA, and if it's done at your home institution, submitting the DNAs to our core or to any other sequencing core that's available. But certainly for the partners, we do all of that sequencing here in-house. And then they receive a big data file of sequences, either sequences of a metabarcode, sorry, I don't know how to shut off that, or sequences of, shotgun sequences from a bacterial genome.
Okay, hold on just a second.
And then those are the basis for the bioinformatics training that's done at the home institutions. And so I'll walk through that with you a little bit, but it's basically first starts off by just developing and establishing those command line skills, and then going through the steps of the analysis that I'll outline in a sec. So those video tutorials are broken up into a couple of different sections. This first section is meant to be, this is the non-active learning, and unfortunately, these are presented by me, and I have been told that they're too long, and all sorts of things. Nevertheless, for people that and students that need some very focused information that will help them understand the data and other things that are to come, like how the data is generated, how the sequencers work, and what the data structures are and conventions, I think that there is some useful information in that. Most people get started at the getting started stage, which is logging onto the server, which turns out to be one of the more challenging things. You know, we'll typically have a class that has, you know, maybe 25 students in it, and they're sent their information, their login and password for the server, and that's usually when we start getting calls about we've never logged into a server. So we have a video that's dedicated specifically to when you get that information, here's how you do it. And then once they're there, these are a whole set of very focused, very short, you know, 5 or 10 minute videos that cover the majority of the core elements of working in this environment. So simply things of, you know, when we are going to run a program, there's a very common set of things that we call the anatomy of a command. So if you're going to run this program, the way that these things are done is not mysterious. At the beginning, it looks like gobbledygook, but they're structured in very obvious ways. And once they learn those, it really opens up a whole world to them. And then there are all kinds of things like you might expect, how to move around in the environment that you're in, how to move and copy and delete things, how to set up an editor that you can do things. Really super important things like grep, which is a tool to go through vast amounts of data and identify individual cases, kind of like a search command. And all the different kinds of things that are useful tools and prepare them to be able to run bioinformatics programs in this environment. And those are all freely available online. You don't even have to know me or anybody. So, and then we go through, for example, I'll use the example of our case where each student or pairs of students, or however you want to do it, it doesn't matter, have the data set from a bacterial genome. So you know, if a bacterial genome is 3 to 5 million base pairs long, they'll have enough coverage to make a very good assembly, meaning they have enough DNA sequences to perform a high quality assembly. They go through managing quality of that data using very commonly used tools in those environments, all run in that bash environment. And then they'll go through and assemble those genomes. So taking all of those individual short DNA sequences and tiling them back together into a genome. And this is one of the first opportunities to understand more about the architecture of a genome and why in the world does the assembly work in some cases and not work in others. And so it's a real rich opportunity to talk about the structure of genomes, even very simple genomes, and then by inference, well, what would happen if this was the genome of a pine tree, for example. And then the other place that where this becomes very locked in and supportive of curricula in courses like genetics or micro or even first-year biology, where you're studying the central dogma. Because when it comes to annotating a bacterial genome, the programs that are used, for example, this one, PROCA, is really just reverse engineering the central dogma. And so you can have these wonderful conversations about, so how do you suspect this program can find a protein coding sequence, which opens up a whole discussion about start codons and stop codons and the genetic code, and they can really see sort of how that works, and they can generate novel data from a novel organism that basically uses that information. And so it removes the mysteriousness of the programs that they're running, and reinforces their knowledge of those biological steps, transcription, translation, etc. Okay. And then they have the opportunity to use those data to do comparative work. Now that they've assembled their own bacterial genome, the first thing, of course, that they want to know is it like anything else? And so we can go through the process using very common tools like BLAST. We can show them, well, so now it looks like these bacteria are all very similar to yours. Why don't we use the command line to automatically download all of those genomes so that we can make those comparisons, and reinforcing how you do that and how you can transfer files. And then we can do large-scale genome-wide comparative analyses using very common tools like OrthoFinder, and then be able to ask questions about, well, why is it that my bacteria has almost all of the same genes as this other one, but it's missing all of the genes for this particular pathway? And what would be the biological consequences of that? So it's an opportunity for some really interesting discussions. So that is the program that we started with, but one of the things that we experienced was a lot of faculty that were sort of in our program were also interested in more environmental sciences, and in particular in the age of microbiome analysis and metabarcoding, we're interested in looking at communities, not just the subset of bacteria that you can do in individual cultures. And so we then introduced to our program a second track, which is basically the microbiome analysis or metabarcoding analysis. So we have a genome assembly one, and then we have the analysis. And that analysis is also done in the command line, and it's done with one of the more popular sets of programs that are used very commonly. So I'll just skip over these. So in this case, part of this was driven by ongoing research, and part of it was driven by cost considerations that we'll talk about in a little bit. But being able to sequence PCR fragments is really inexpensive, and the analysis is fairly straightforward. And all of this is done in QIIME 2, and we have additional support tools for that as well. Oops. And so, so when we're talking about barcodes, I think the ones that people are most familiar with are things like 16S ribosomal RNA, But also depending upon what kind of organisms, if you're working on more on animals, a lot of people are using mitochondrial genome fragments. So if you're doing environmental DNA, those are very popular systems to work in. And so we support all of those. The next thing to talk about in terms of the support is the computer and IT support. So we have a server that's called Ron that has approximately, I want to say 2,000 users on it. Ron is a fully enabled high-powered bioinformatics server. It is specifically dedicated to training. However, we allow people to stay on there and complete projects. We have another set of servers that we migrate people over to when they become in need of that sort of thing. So this is a nice opportunity. We did this on purpose, and so this is a server that we can control ourselves. Because if you've worked on a server and in many of the other environments, you know that you sometimes everything will get slowed down because somebody else is running a whole bunch of big jobs, and so we have complete control and can shut people down and turn people back on for this. So when we happen to be running a workshop that's trying to do RNA-seq on 50 people, we can monitor the environment and do that. This hosts a full suite of open source software, and any of which, as long as it's open source, can be added at any user's request. And as I mentioned earlier, I think the biggest issue that we run into here is just people logging in. It only takes about 1 minute to forget your login credentials, and that seems to be the most common issue. So the other support is sequencing, and so this is a little bit of an old slide, but I think the sequencing costs and even the library costs are dramatically reducing. But just to put it in perspective, these samples are, even a whole publishable bacterial genome is less than $100. And so it's certainly not out of the range for a lot of people to use. And I'll talk about other ways of doing this. And for the metabarcoding, I forget what it is. Something about, I seem to remember the last time I told somebody it was like 10 million reads for 25 cents or something like that. The critical issues with implementation that we have come across. The most important one is that at any institution, the average faculty member this is not core skills that they have. It's not something that they brought to the job necessarily, and so they're not experts. And most of us have the opportunity to teach in areas where we're experts.
Hey guys, I'm Dave Batista.
It's really hard to teach things that you don't know. I think we can agree on that. So this is where we decided to provide some focused help and led to our IPERT program, the Train the Trainers program, and also led, at least for the support of our partners, to the need to have really focused help for those faculties through the first few iterations of their classes. The other thing, I don't know if I'll talk about it again, is we're talking about a couple of case studies that are primarily microbial, and not everyone teaching genetics, for example, is a microbiologist, and so they don't have necessarily the skills or a bunch of bacteria or even environmental samples available to support their class. And so what we have done over time is develop relationships with some fairly large scale research groups that have unlimited numbers of samples and love nothing more than to donate them to classes. And the classes then, as I'll talk about, can go through those processes and develop novel genome sequences or novel community analyses that can then be published, and both the class students in the class and the PI of the lab that donated them can be authors on those. So just to give a plug for Train the Trainers, this is where we hold Train the Trainers when we're not in COVID lockdown. This is the Mount Desert Island Biological Laboratories. The next iteration of this is hopefully June 26th through July 3rd. It will be those dates. It's hopefully that we will be there. I don't know if I don't have a pointer necessarily, but the building to the right center that has a patio, a red patio that looks out over the water. The downstairs is where we do the molecular biology parts, and the upstairs is the bioinformatics suite. And that is where we take our breaks. And it has one of the best views that you could possibly imagine. It is a beautiful environment, and I strongly encourage you to come and join us if you can. The program is a NIH IPERT program. It's essentially fully funded, and so get your applications in as soon as you can. And I hope we're there in person. So I don't labor the points too much, but you know, for the microbial genomics, the case studies that have become very common for us are things like antimicrobial compound discovery. There's lots of labs, there's lots of things to talk about there. One can do those yourself with things like crowded plate assays that you can have interesting biological discussions. We also have a large number of streptomyces colonies that are available from research groups where more than, you know, the vast majority of our antibiotics are derived. This year in particular, we focused on sequence analysis for SARS-CoV-2, where there's they could implement some of their comparative genomic things at a large scale on those. Things like biofilm formation, very important in certain diseases, and can be analyzed using natural isolates of Pseudomonas. And then simply interesting, there's an endless number of these interesting projects that people have done, like sequencing the snow seeding microbes, which I guess I knew that they did that, but you know, sampling snow after a seeding process and culturing out those bacteria and sequencing them to see what they are. Similarly, in the microbiomes, there's a lot of interest in people that we work with on working in what's called the built environment, and you know, like your workplace, your home. It's always fun to go to a brewery if you're of age, and sample those things. Tracking human and animal waste, and one of the ones that we had the most fun with was tracking the feeding habits or pollinating habits of things like bees, where you can sequence the pollen grains or using a metabarcoding approach and figuring out what species they visited. The final thing I want to touch on, excuse me for a second, and sorry if I'm running a little overtime here, is publishing. And so one of the most, I don't know, positive experience that I had was within our INBRE program, simply having the students be able to submit high quality genome assemblies to NCBI that has their name on them, and it's permanent, and they can go on the NCBI site and see their name and their contribution. I think it's really been impactful on them. And to the ones that you get to meet their parents, this is what they told their parents about their bioinformatics experience. And there are all kinds of levels of publishing, of course. There's most of the students that we work with, the primary goal is to get those sequences into GenBank like this. And so this is one of our partners at New England College and one of the groups that they've worked on. And then there's also groups that have taken this to an actual publication for things like genome announcements. And we have some fairly prolific groups that seems to be the endpoint for every one of their classes, for example. So I want to stop there because I think I've rambled a bit, and just thank, a lot of the development of this really was built on the backs of our faculty at our partner institutions, and then of course my own group that did a lot of the work and actually deliver this in most cases. So thank you.